animals, specifically mice, because we're trying to understand how they cause disease. And what we found was something that was sort of a simple observation, but we thought might be useful to know. This is our old friend in the lab, Mycobacterium tuberculosis. This is the thing that causes TB. 2 billion infected, 8 million new cases globally in the world. And when you put tuberculosis into an animal and you look at it under a 400-fold microscope, you see these little things there. Those little rods are tuberculosis. And when you use an oil microscope, which is 1,000 times magnification, you see those three. The same three are those three, okay? So you sort of see the pattern of those three. And if you use pixel integration software, you can ask this computer to cut out little things. So we have this set up so that we have lone bacilli that are alone. And then you ask the software, how many pixels did that bacteria cover? And it says these bacteria are about 4 microns long and about 1 micron across. What that means pragmatically is if you multiply it 1,000-fold, it looks about like a 4 millimeter thing. So you imagine the ruler you have. If you have 1,000-fold magnification, you got that thing that's like that, okay? Just over a centimeter. Well, here's Mycobacterium avium paratuberculosis. Under a 400-time magnification, you see that little thing there. And most of us would say, well, maybe it's a bug, maybe it isn't. It doesn't really look all that distinctive. And even at 1,000-fold magnification, that bug is this one here. There was a little thing there that now is popping up, and there was a little thing up there that is popping up. Even at 1,000-fold, these are not actually distinctive, long, skinny rods that jump out at you. In fact, our eyes are trained to see a stick better than a blob. It's just the way we are. And when we asked the software how big are they, they said, oh, it's about 1.5 microns, which means if you multiply it 1,000-fold, it's 1.5 millimeters. So if anybody has a ruler, take a look at a ruler and say, okay, now I'm trying to see if there's a 1.5 millimeter thing there. So why does that matter? Well, it turns out that this 400 is what pathologists use, and this 1,000 is what microbiologists use. So in 75 years, all we had seen when we read every article from 1932 to 2007 was this one. We had never seen anybody use a 1,000-fold microscope. So we said, let's try it. Let's use one of those microscopes and see what happens. So we got people with Crohn's disease and controls. We asked them, do you mind if we go back to the surgery you had in the past? We're going to study the tissue that's already in the path lab, okay? And we put it under a microscope with 2 stains and looked at 1,000-fold. It was done at 2 sites, with 2 readers, so Montreal, Hamilton, 2 readers, 1 at each site. And it was done blinded so that the person in Hamilton had the code for the Hamilton people, I had the code for the Montreal people. All the staining, all the reading was done without knowing who had what disease. Now, as you saw in the previous picture, we're looking for little blobs. So we're not expecting perfection, okay? We're just trying to ask the question, are there little blobs in Crohn's tissue? And this is what we came up with, okay? So we said here, there's 100% there. And Crohn's is in white, and controls, and that was a mix of ulcerative colitis, diverticulitis, cancer, and occasionally volvulus, they're in gray. And in Montreal, half of the Crohn's patients, 50%, were positive by both stains. Stain 1, stain 2, positive. And in Hamilton, 60% of the patients with Crohn's disease were positive by both stains. In Montreal, about 50% of the controls we said were doubly negative, and in Hamilton, about 80% of the controls were doubly negative. Now, so this isn't perfect, right? We're not just black and white here, we have a sort of a trend. But for instance, here in Montreal, there were a couple of people who were controls who were doubly positive. And as people in the room may know, and people like Mark will know, you look at the chart, and it says ulcerative colitis, and then they recurred, and they had a colectomy, and they recurred, and there was disease in the terminal ileum, but we said, the chart says ulcerative colitis, we're going to keep them ulcerative colitis. Maybe they had Crohn's, I don't know, but we left them at that. But nonetheless, if we ask, how common was it to be doubly positive for Crohn's compared to the controls, we got a risk of 8.5-fold. So it looked like in the tissue, Crohn's tissue was 8.5 times more likely to have these little bacteria sitting in them if you looked hard enough. Okay, so that's nice. So there's an association, but as I mentioned, now we're working backwards. Did we know that the bugs were there at the beginning of the disease, or did the bugs get in there during the course of disease progression? We can say now that Mycobacterium avium paratuberculosis is found in Crohn's tissue at the time of disease, often at the time of surgery. It's been done by the PCR by many groups and many labs around the world, and was done by microscopy by one study. And I'm waiting to see if somebody either replicates or refutes our studies, because all good science should be reproducible independently by another group. But we still are left with the question, is the organism a bystander? Is this organism we're seeing the thing that started the problem, or is this organism something that piled on after the problem and is a secondary contributor? This study architecture cannot assess that. All it says is it's there. Okay? And so we've written up a piece on an essay on the difference between association and causality that came out this week in a journal called Lancet Infectious Diseases. So if anybody is interested in understanding those differences, that's online, or if you send me an email, I can send you the PDF. So, known unknown number 2. Am I going way over time? No, we're still okay. Is Crohn's a genetic disease? Of course it's a genetic disease, right? Twin studies said so, ethnic associations, linkage analysis, whole genome associations. Crohn's has been at the forefront of genetics. There's been fantastic advances in Crohn's and understanding the genetic basis of disease. So now you're asking yourself, well, this guy's telling me there might be a bacteria. Is there a way to reconcile this? Well, it just so happens that every Friday afternoon I have lab meetings with a guy who studies leprosy. And he tells me leprosy is a genetic disease. Because if I go to his lab meeting, he says there's twin studies, linkage analysis, all those things you just saw on the last slide. I just copied the slide, right? Using the same types of studies, they told us that leprosy is a genetic disease. And why is that? Well, leprosy is caused by a bacteria. In fact, it's the same genus, Mycobacterium leprae. And why is it a genetic disease? Because he says it's a genetic predisposition to a bacterial infection. Okay? So if Crohn's is a genetic disease, that doesn't mean it can't be a mycobacterial infection or an E. coli infection. Now we have to do some more work to understand what kind of interaction there might be. So, you've probably heard earlier today a bit about this. What are the Crohn's genes? And as I simplify it, I think there are a number of genes that have been associated with IBD. And it might be Crohn's or colitis in certain studies. But right now, the ones that I think have been really uniquely and specifically associated with Crohn's disease are NOD2, autophagy 16, and IRGM. But Mark, you can correct me if I'm wrong, those are the 3 that Judy Cho put in her latest review as associated with Crohn's disease and not ulcerative colitis. So what are we doing about that? Okay, we start to think about this. We're bacteriologists, but we're trying to understand how the host sees our bacteria. So NOD2 is the first Crohn's susceptibility gene. If somebody harbors 2 mutant forms of the gene, maybe a 30, 40-fold risk of disease. 1 copy, maybe a 2 to 4-fold risk of disease. It is known that NOD2 belongs to a family of proteins that work as intracellular bacteria sentinels. They recognize bacteria inside the host cell. And in fact, it is known from 2 groups that the thing it recognizes is the bacterial cell wall, and specifically a chemical called muramyl dipeptide that is in the bacterial wall. We know that people with Crohn's mutations have decreased recognition of this MDP, so we asked ourselves in the lab, how could we try to understand this in an experimental setting? Okay, so it turns out that if NOD2 recognizes the bacteria, and Crohn's mutations have decreased recognition, that if you go and obtain a mouse that is a NOD2 mutant, where somebody has used slick mouse engineering to knock out the mouse NOD2, that mouse has the same defect in recognizing bacteria. As I've told you, we cannot take people who have these mutations and feed them things and wait to see what happens, but we can take mice that have the mutation and see what happens. We can ask how do they feel about having a mycobacterial infection. So, at first you'd say, okay, good, why don't you put Mycobacterium paratuberculosis in the NOD2 knockout mouse, Dr. Behr, and tell us what you found. So we looked around in literature and there was no kind of published model on putting Mycobacterium paratuberculosis into mice.